Hello, we are delighted you've joined us for today's webinar, NGS Live in Your Lab in 30 Days. I am Christina Mahalik of Labroots, and I'll be moderating this session. Today's educational web seminar is presented by Labroots, the leading scientific social networking website and provider of virtual events and webinars. Today's webinar is sponsored by QIAGEN. QIAGEN, a Netherlands holding company, is the leading global provider of sample assays, technologies that are used to transform biological materials into valuable molecular information. Sample technologies are used to isolate and process DNA, RNA, and proteins from biological samples such as blood or tissue. Assay technologies are then used to make these isolated biomolecules visible and ready for interpretation. QIAGEN markets more than 500 products around the world, selling both consumable kits and automation systems to customers through 4 customer classes: molecular diagnostics, human healthcare; applied testing, forensics, veterinary testing, and food safety; pharma, pharmaceutical and biotechnology companies; and academia, life science research. Let's get started. You can post questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them in the Q&A box, which will open when you click on the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at the lower left of the screen. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I now present today's speakers, Yi Kong, PhD, and Salim Esakali. Yi is currently a product director at QIAGEN, responsible for content and application development of cancer molecular research and diagnostics. She led efforts to develop and commercialize the GeneReader NGS System. She has years of experience working in the diagnostic and pharmaceutical industries in various development, product management, marketing, and strategic planning roles. She started her career as a biomedical student, having published many peer-reviewed articles and a diagnostic patent. It is my pleasure to introduce Dr. Kong. Dr. Kong, the floor is yours.
Thank you, Christina, for that generous introduction. Thank you, everyone, for taking time out of your days to attend our webinar here. Today, my colleagues Salim Esakali and myself would like to present to you the GeneReader NGS system and tell you a little bit about how your lab can go live with NGS in 30 days. We have structured today's presentation as follows. I will begin by telling you a little bit more around the concept of live in 30 days. And then my colleague Salim will introduce some more information on the GeneReader NGS system. Specifically for NGS live in 30 days, I would like to present to you some of the challenges to implementing an NGS workflow and explain how the GeneReader system is different. I would also like to share some of the data we have generated internally at the QIAGEN, as well as externally in collaboration with some customers. So as many of you already know, NGS has proven to be a very powerful tool in understanding the complex genetics behind a tumor or cancer. But there are also many barriers to a broader adoption of this fantastic technology, owing to a number of factors. For example, the current solutions on the market are fragmented and the workflow needs custom assembly of various components from various vendors. Second, while sequencing is relatively easy, the challenge of analyzing and interpreting the data can sometimes be very daunting to a laboratory, especially one that does not have a lot of bioinformatics experience. And last but not least, the cost of the NGS run can be quite high and obscure to a laboratory. And this is exactly why we developed the GeneReader system by offering an integrated workflow and by also offering a seamlessly integrated bioinformatics solution as part of the solution, and last but not least, offering a true transparent cost structure to ensure its cost effectiveness. Typically in a laboratory, the process to bring up an NGS workflow takes around 6 to 9 months. This is because a laboratory begins by choosing a sequencing platform first, and then the lab has to choose an upstream sample preparation platform, and then has to find a QC solution to the workflow, as well as a bioinformatics solution to it. And then the laboratory needs to design and perform its own verification assay to ensure that all components work together. And if they don't, the lab has to conduct troubleshooting and optimization processes. And this process goes on until the laboratory obtains a satisfactory result and can then go live with an NGS test. And this is typically a 6 to 9 month process. But for the GeneReader system, once we install the system, because it comes with all the components that are needed, the laboratory is ready to perform a verification process with the protocol and expected results that also provided as part of the system. The laboratory can then go live with NGS in as few as 30 days. And this is because the NGS process is inherently complex and spans multiple days. It goes from nucleic acid extraction to target enrichment to library preparation to sequencing itself, and then to data analysis and interpretation. And the GeneReader NGS system comes with all the automation platforms, all the hardware, software, as well as reagents to cover each and every step of this complex process. In addition, we also offer several QC platforms that can be used in this process to make sure that every step of the way is performed up to the expectation. Moreover, every GeneReader system comes with an iPad and an app called GeneRead Assistant. This is an app that takes the place of a traditional handbook and provides an interactive and simplistic user interface to allow the laboratory to follow the protocol and control the operation of the workflow. Last but not least, the GeneReader also comes with a GeneRead Link software to specifically allow bidirectional communication between the NGS workflow and the laboratory information management system, thus allowing the laboratory full visibility, full control, and chain of custody for the entire NGS process. And this system is available in the United States with a brand new chemistry. And what do we mean by full protocol and full control? And what does a laboratory need to do in order to test and go live with the NGS with the NGS operation? So as you saw before, there are multiple steps in this whole process to get to an interpretation or interpreted report. And what we've done on the bottom of the slide is by combining this entire system with a rigorous testing of a set of Horizon reference standards to provide a fully integrated solution with a full process control and a very robust set of performance data that the laboratory can use to measure against and to guide their setup of the workflow. Just to give you an example of the results that you can expect, this is a set of data obtained from FFPE reference standards from Horizon. You can see the list of genes and variants on the left-hand side with the expected allele frequencies in the middle of the graph with a lower and higher range. As you can see highlighted in red, the allele frequencies obtained on the GeneReader system very closely align with the ranges, with a very tight standard deviation next to it. And this is obtained on the average of 10-plex coverage. And as you can see, we have pretty uniformly high coverage across the board. This is perhaps better represented in a graphical manner as shown here, where each gene and variant is listed on the bottom in the x-axis, and the variant frequency is listed on the y-axis. And each single dot represents a separate experiment in which we vary a number of different parameters, either the site at which the experiment is conducted, the operator, the sample batch or reagent batch, and the level of plexing that we apply to that particular experiment. And again, as you can see, regardless of the variables, you obtain a very robust and consistent level of performance across the board. This is just to give you a reference that Palmetto GBA's recommendation in NGS operation. It recommends a reproducibility study covering exactly all the parameters that we just went through in terms of operator, in terms of runs and batches, in terms of sites, and the reagent lots. So this is the process that laboratories typically go through, and this is exactly the process that we at QIAGEN have carried out. So one of our collaborators and customers, Dr. Yasmina Curry at Legacy Health Networks in Oregon, has been using the GeneReader since 2016, and in her experience, this kind of data provides a very robust and good reference standard for her to set up an NGS operation. And this data set is the first of its kind. At the very end of this hour, you will also be able to watch a very brief video interview of Dr. Curry, in which she will share her experience using the system as well. This slide is a set of results tested from liquid biopsy samples at a lower allele frequency of 1%. Again, you see the list of genes and variants on the left-hand side with the expected allele frequency in the middle and the standard deviation or the lower and upper bound around it. Again, highlighted in red, you see the observed allele frequencies on the GeneReader to be very closely aligned with the reference standards. And again, we'll have a uniformly high coverage across all the genes and variants. And this is again meeting or exceeding the recommendation circulating tumor DNA testing as published by Palmetto GBA. Next, I would like to show you two pieces of studies that we are publishing, the process of publishing in collaboration with some other sites. This is with Dartmouth University, and we conducted a concordance study between the GeneReader and another NGS platform. And again, as you can see, each dot represents a single variant with the allele frequencies plotted out based on GeneReader output as well as the alternative NGS platform output. And again, as you can see, from very low to very high allele frequency range, there's a very close correlation between the two platforms. And this is replicated with another study conducted with University of Cologne on another alternative NGS platform. We also have a number of abstracts published at various conferences and trade shows. To highlight one of them, which is AMP Global, which is taking place in Berlin in April of this year. We have 7 abstracts accepted there. So if any of you will be joining us in Berlin, I welcome you to come to our posters as well as our workshop and booth. I would like to end my part of this presentation by also sharing some experience from Professor Kurt Talupa at University of Graz in Austria. He has also been one of our early adopters of the GeneReader system, and he sees a lot of value in a laboratory, in one vendor providing a total solution for the entire NGS workflow for laboratory to enable a much faster testing process and a laboratory to go live in as few as 30 days. So I would like to end here and share a very brief video from Dr. Talupa sharing his experience, and then I will pass the floor to my colleague, Salim. Thank you.
My research group mainly focused on bringing new technologies into medicine. You cannot sufficiently predict the behavior of a disease. So we need deeper insight into the molecular biology underlying a disease. And that's where sequencing and molecular testing comes into play. We had to learn that a single alterated parameter is not able to describe an individual disease because it's influenced by so many different parameters. And now, thanks to next generation sequencing, we are able to get a much more comprehensive view on the variety of mechanisms underlying disease. We learned about the importance of a complete integrated workflow. If you compile your analytical process by different products and technologies from different companies, you never know how they fit together. And to have reliable results at the end, you have to validate the whole workflow. And therefore, we are so in favor of the approach of QIAGEN, doing this already on an industrial scale, so the workload is not on the user's level. A major underestimation is that at the moment people just calculate the costs of an analysis, not considering the costs of validation of a complete process. And there I expect a major additional benefit by a concept that essentially provides a fully integrated workflow. And then this integrated data analytics pipeline based on the Ingenuity technology is so key for the proper interpretation and drawing the right conclusions, because it's not just producing NGS data, it's really doing the data interpretation, which is also recognized as a major bottleneck in this field. At the moment, I'm not aware of another product like the QIAGEN NGS workflow. It's not just the GeneReader, it's the whole workflow. So therefore, I'm very convinced that this is the best way to go.
Informative presentation. Next, it is my pleasure. Thank you, Dr. Kong, for that informative presentation. Next, it is my pleasure to introduce Salim Essakali. Salim Essakali studied biology at Heinrich-Heine University, Dusseldorf, Germany, and specialized in biomedicine. He conducted his master's thesis at Peter MacCallum Cancer Center in Melbourne, Australia, analyzing gene expression profiles in chronic lymphocytic leukemia. In October 2007, he joined the global product management team at QIAGEN in Hilden, Germany, and has been responsible for managing multiple automated platforms in bioinformatics solutions. Mr. Essakali, the floor is yours.
Thank you very much. So I hope you can see the slide here, and thanks for this very nice introduction. And so I would like actually to now just briefly reiterate how the GeneReader NGS system actually enables to be up and running after only 30 days. And there's actually 5 design criteria which I would like to explain here. Before, though, I would like to share this slide here. So basically we started actually with a question first, and that question was, what is the ultimate objective of next generation sequencing? And as you could see already also within the slides that Dr. Kong has shown before, the ultimate objective that was identified after speaking to all of these labs that are experts in next generation sequencing was this report here. So to end up with an actionable report at the end of the NGS workflow. So to take all of this data that next generation sequencing can actually generate and only end up with a 1 or 2 pager that shows the most actionable information only. And to reach that objective, actually there was 5 design criteria identified. The first one is to be as complete as possible. Actually, Dr. Kong has shown that already. But I would like to just revisit this point very briefly. The second one is to be as actionable as possible. The third one is to be as flexible as possible with multiple flow cells that can be sequenced in parallel so that the lab can scale up and down depending on how many samples need to be processed. And then the fourth and the fifth go back to the first one. So because this solution is supposed to be as complete as possible, also there was the design criteria then to use this completeness and make the pricing also as complete as possible, as transparent as possible, but also as flexible as possible. And then the last one is service, so to have a holistic service along the entire workflow as well. So what does truly complete mean? You have seen that slide already before, so I don't want to spend too much time on that. So it means not only that we cover the entire workflow, so extraction, target enrichment, library prep, sequencing, data analysis, and interpretation, but also the respective automation, and then very importantly, as was highlighted before, quality control, and then user support. You actually could see the user support tool in the video before, the iPad app that really guides the user through the entire workflow so that the user wouldn't have to worry about user documentation anymore. And then last but not least, data management. So this is actually a very powerful tool that shows on one single screen the entire workflow in terms of how many samples do I have in my laboratory that are currently in extraction, how many of these samples, and actually also which samples have passed the QC or maybe failed the QC and had to be reprocessed, and this for the entire workflow. So how many samples at library prep and sequencing and which of these and what happened at the QC here. Very powerful also then for LIMS integration later on. So actually it's almost like a laboratory information management system from sample to insight that only needs a LIMS in and LIMS out configuration, and ready is the LIMS integration. So this is the completeness of the entire workflow, and I just wanted to highlight actually that every assay that has been developed for the GeneReader NGS system undergoes this entire workflow in terms of development and harmonization to really achieve the results that were shown before by Dr. Kong, and especially to achieve or to enable this report in the end, and especially to enable this in 30 days. So this is the completeness, and actually just to summarize this very briefly, so the completeness really is addressing or avoiding the pain of harmonizing the entire upstream solutions. It also avoids the pain of maybe even having to build a bioinformatics department from scratch, and really is about realizing a true solution. So not just to have 1 or 2 parts of the workflow covered, but really a true solution from sample to insight, as we say, that has been fully harmonized before it actually leaves our research and development and our stock towards a lab. The second design criteria is to be really focused only on the most actionable insights, and to do this actually I'm going to start with this slide here, to do this in a way that is not only, let's say, holistic, but also at the same time is at the state of the art in terms of medical and scientific knowledge. We go back actually to this QIAGEN knowledge base, which has been built over almost the last 2 decades. So in there is over 13 million findings, as you can see here, over 20,000 disease classes, over 30 different databases, over 30,000 publications, and over 340,000 samples that have been analyzed. And this database is then used, by the way, this database is updated on a weekly basis, and this database is used to design the panels in the first place, to make sure that really only the most actionable insights are being covered here, to not drown in data later on, which is not actionable, and therefore get the most out of the system and the technology. And actionability in this case has been geared towards either a treatment option, clinical trials, or professional guidelines. And for the first panel that has been launched on the GeneReader NGS system, the Actionable Insights Tumor Panel, that knowledge base has been used to query that knowledge base for the solid tumors with the highest prevalence, the greatest need for testing, which means that these solid tumors have the most variants actually which are associated with actionable insights. And you can see in the left-hand bubble, actually the results out of that query were the big 5 solid tumors, so no huge surprise here with melanoma, colorectal, lung, ovarian, and breast, and 12 genes that are covered with this Actionable Insights Tumor Panel. So it's a very powerful first-line testing for solid tumor panels. This slide here actually shows the footprint of the panel and some other parameters such as the insight sizes, the amplicons, amplicon size, DNA input, throughput, and variant frequency and amplicon coverage and variant insight coverage, and also compares these parameters to, let's say, similar solutions that are out there. And what can be seen here is certainly the throughput is one parameter where the Actionable Insights Tumor Panel gives the highest flexibility in terms of samples per run, but then also the amplicon coverage, as you can see here, is very high, and the variant insight coverage is also very high. We have also analyzed this sample, this assay, the Actionable Insights Tumor Panel, in terms of actionable insights or actionable variants which are actually being missed by similar solutions out there. So this is just due to the fact of that knowledge base that we are using for the design in the very first place, which is highly geared towards actionable insights only. And matter of fact, what you can see here is on the bottom, the total is the number of variants, actionable variants, which are missed by other solutions. So you can see here 180 are being missed, 117. And even if you would combine these two assays together, you would still be missing around 80 actionable variants. So again, this is just showing the design criteria of actionability that is behind this with the knowledge base. That knowledge base allows later in the bioinformatics. So actually, I should actually start with the analysis first. The analysis is very straightforward. It has been actually, again, as the entire workflow, highly optimized towards the application at hand. And this is basically a push-button solution, but at the same time allows maximum visibility in terms of if someone would like to review some of the variants manually, this is very nicely doable in the analysis tool. Then the next tool, which we see on this slide here, is the actual interpretation tool. And So what it does is it doesn't only help to directly create the report. So the degree of automation is definitely maximized, but at the same time, we also have a maximum of flexibility too. So in other words, what you can see on the screen here is before the report is being created, one can review the interpretation of a variant at hand. So you can see here on top some further details with respect to that variant, and also below whether it's pathogenic or not, and then below that you even see the missense effect on the protein. I hope it's actually better readable on your side than on my side here. And then even the treatment information can be selected here and set in terms of the evidence level as well. So it is really a combination of high degree of automation to get that report, but at the same time it's very flexible too if one would like to deep dive into it, and also very customizable as well, and very transparent too. So all of the information, where this interpretation is coming from, which publication this has been drawn from, or which clinical trial, or which professional guideline, all of that information is in here as well. So it's a very powerful tool for the interpretation to create that report. So that was the Actionable Insights tumor panel. What we are launching right now is also a Bracha Panel, which is targeting both breast and ovarian, and also both mutation types, germline or somatic, and is highly geared towards clinical actionability of PARP inhibitors. And the next slide is actually just briefly showing the design criteria for that Bracha assay. So we are covering the entire exons plus 20 base pairs flanking on each side, and the small indels go below 6 base pairs, and then large indels reach to 40, down to a sensitivity of 5%, the specificity above 90%, and the amplicon coverage here, as you can see, is very high as well. So this is the QIAGEN Bracha Panel, and again, as I said before, every assay is not just harmonized in terms of that assay itself, but really is embedded into this entire workflow, and undergoes heavy development and harmonization before it's being released. And that includes, again, of course, the bioinformatics as well. This is just one screenshot showing the interpretation tool here again for Bracha, and here you can see how it's being highlighted, either in red if it's pathogenic or green if it's benign, and then you can go through that and actually do the same settings as we have seen before. So here again, the treatment information can be set in terms of the evidence level, and then one can go through the different hits that this knowledge base had in terms of publications with respect to treatments, treatment options, or clinical trials. Then I would also like to briefly show you another panel which we are currently launching, which is the QIAGEN Lung Panel. And as you may know very well, lung is an interesting use case here, especially because there's so many different variants that need to be detected, and some of them are really tricky too, such as the point mutation here at EGFR, large deletions like the MET exon 14 or the ALK-CNV or fusions, and the left-hand side here are really just examples, and by no means the entire content of the lung panel, but just examples of, let's say, tricky mutation types that needs to be detected in lung cancer. And on the right-hand side, you can see how many different technologies usually are being utilized to cover all of these different mutation types. And the technologies reach from PCR, IHC, up to FISH and arrays. And this is especially an interesting case, let's say, where next-generation sequencing can help to drive or to offer one single assay that includes all of these results. And that's exactly the aim of the lung of the QIAGEN long panel, as you can see on this slide here. So it's geared towards 100% actionability. You can see here with around 550 mutations covering all sorts of mutation types. So whether it's SNVs, small or large indels, CNVs, fusions, and then the sensitivity you can see here goes down to 5% for FFPE or below 1% for liquid biopsy. The specificity again is above 90%, and then the variant coverage is also very high to reach all of these specifications here. One thing I would like to highlight here, especially with respect to the lung panel and all of the other assays that are developed for the GeneReader NGS system, is the unique molecular indices, a technology actually which uses, again, unique molecular indices or UMIs to tag original DNA molecules at the very beginning of the library prep or after extraction, so to say, to be able to identify those original molecules. And the idea behind that is very straightforward. You can see this here in the two cases. Once we detect a variant, then after library prep and after target enrichment, especially after PCR amplification, then this UMI technology can be used to strengthen the statistical significance of a false positive or a true positive. On the left-hand side, we see the case of a false positive on the left-hand bottom. And there you can see that there was the star means the mutation, and you can see that that mutation seems to have occurred only in one of the original molecules, and all of the other copies of that original molecule actually don't show that variant. So this is showing that very, very likely that this is a false positive, and it can be very nicely identified then with strong statistical significance. While on the right-hand side, same case, but in this case it's actually a true positive, and of course by having those unique molecular indices, and one can say, okay, each and every of these original molecules actually had this mutation. Of course, this strengthens this detection of a true positive by multiple magnitudes. And actually, the magnitude of that statistical significance is shown on this slide here. So we asked our bioinformaticians actually to analyze and translate this into the Q-score. And here you can see in this graph the Q-score before taking that UMI technology into account, and you can see a standard distribution around Q-score of 30-ish, so a very normal standard distribution here. And then after the UMI technology has been taken into account, so after all of these stacks of molecules, it can be traced down to the same original molecule, and the significance or the statistical power of that technology can then be seen by this huge jump in the thread score, which actually had to be cut at 60, just because, even 60 is probably tough as a number to be, let's say, experimentally verified. And that's why actually this cutoff had been taken at 60. It's just showing actually what a specific assay design can actually do if it's combined with the right bioinformatic analysis. Or the other way around, actually, it's just strengthening again this completeness strategy to make sure that it's not just about one or two components along the workflow which needs to work nicely, but all of these components together, if this is orchestrated the right way, you can even leave the same sequencer in the middle here, as you can see with this slide here, just by actually controlling the assay design in a specific way and the bioinformatics in a specific way, and make sure that everything is harmonized, can have this huge jump here in terms of performance. So it's just another proof point actually for this completeness factor, which again is in the end helping or geared towards that report in the end, and especially to enable that report within 30 days. This slide here I actually stole, so to say, from our R&D department. So it's just showing actually the development practice and when something is actually being released. So I thought I'd just show this here because it just shows the due diligence that goes into these assay developments. And again, so maybe I'll just briefly just explain. So you can see here experiments, instruments, users, sites, and then different categories of, let's say, statistical significance again. And of course, there's tons and tons of experiments behind that. But what I would like to highlight here is that each experiment or each of these levels here, again, always means sample to insight, so that really the entire workflow has been tested through. Tons of experiments, sample to insight. Same for the instrumentation, so multiple different, definitely lots of different instrumental setups that are being tested, sample to insight. Again, also for the users, sample to insight, and sites as well, sample to insights. And only when all of these things come together and have been reproduced multiple times along this entire workflow, and I should actually, again, note here, sample to insight here does not only mean the automation, but including the QC, including that user support tool, and including GeneRead Link as well, the laboratory information management system setup. So only when all of these things come together, and then the subsystems are being, or subsystem, these results are being reviewed on the subsystem level, and then being reviewed again on the system level, only then these assays or these products actually are being released. So just to summarize this very briefly, so these the design towards actionability help really to focus only on the most actionable insights, and enables actually to cover hundreds of actionable insights which other solutions are missing, and therefore also allow then this custom report in the end, and all of this together with highest amplicon coverage and batching flexibility. So this was the second point, and I can actually see already Q&As coming in, which I highly welcome. So we all look forward to the Q&A session, which is only a few slides away, by the way. So the third point, the flexibility, is actually something that is rather straightforward. I don't know whether this slide will translate because it's animated, but so basically the main feature here is that the GeneReader NGS system, or the GeneReader sequencer itself, has this turntable at its core, and that turntable actually allows true parallelization of multiple flow cells. So in other words, we can sequence up to 3 flow cells or anything in between, and whether 1 flow cell or 3 flow cells are being sequenced does not make a difference to the actual sequencing turnaround time, because while the 1 flow cell is actually being sequenced, the other ones are undergoing liquid handling, so to say, incubation, and then as soon as 1 flow cell has been actually sequenced, the next flow cell is being lifted towards the camera to undergo sequencing as well. And this just allows, there's the summary slide missing, so this just allows the sequencer to scale up and down, just depending on how many samples come in. One can then choose whether it's the 1 flow cell or the 2 flow cell way, and therefore the sequencer, the GeneReader NGS system, scales up and down together with the number of samples that need to be processed. Point number 4 is going back to the completeness of the entire workflow. So because the entire workflow really takes everything into account, including QC, etc., we can give full cost transparency here too, which sounds trivial, but very often if a workflow is fragmented, there's multiple SKUs, double-digit of SKUs involved, and just accounting-wise, this is actually becoming a challenge. And here we can give full cost transparency, which was a very important design criteria actually, and then also have 3 different pricing models The one is the CapEx purchasing option. Then there is a reagent rental, and then there is also pay-per insight, which actually allows us even to share some of the operational risks that NGS still includes. So this is basically mainly the number of samples that are being put on a flow cell. If the number of samples is going up or down, this will directly affect the price per sample, and we can use this price per insight actually to regulate that and to share some of that risk with the lab. Okay, and so with this, I'm actually at the last point, which is the holistic service. And this again goes back actually to the completeness of the workflow. So so again, that sounds trivial, but that was very important also for NGS labs when we were interviewing them, that during troubleshooting or whatever, that it's actually one contact that can be used here to troubleshoot, but also to initialize and operationalize an entire laboratory. And there's many cutting-edge solutions here, from application training to consulting, remote diagnostic, that we can offer here with the GeneReader NGS system. And with this, I would like to actually just end with this slide here, which is just showing. So again, just to summarize, so the design criteria were to be truly complete, as actionable as possible, as flexible as possible with the multiple flow cells, to offer guaranteed results with predictable cost, and have this one service contact. And actually now with the new panels that we are launching now, and the detection of CNVs, infusions, and the unique molecular indices, we're strengthening this actionability very significantly, and then also increase the financial attractiveness, which is point number 4. And by also twisting the system or increasing some system efficiency, we were enabling to increase the number of samples that can be put on a flow cell, and therefore actually allow the entire system to be more efficient. And all of these things, again, are geared towards this one objective, or to enable this one objective, which is the actionable report in the end, and enable this especially within 30 days from installation. So with this, I am at the end of my slides, and I would actually just briefly check whether we have the time to just show very briefly the video from Dr. Akari's laboratory and her user experience, and then open it up for the Q&A, which I can see are already coming in here.
Cancer is a genetic disease. It messes up the DNA sequence, and we need to know everything that goes on. We want to be able to offer that kind of testing, being able to look at several genes at the same time, and next generation sequencing is the only technology available to do that. NGS is a very complex technology, and it has several elements, from the DNA extraction all the way to the library preparation, then to the sequencing, and then the bioinformatics, all the way to reporting. I think there's a huge need for a one workflow process in NGS. The NGS system that QIAGEN offers is end-to-end solution in one workflow. It really has no competition out there, honestly. It empowered us, which we loved, right? We loved the idea that we can do something very complex in a very simple manner, simple, cheaper, higher quality manner, and that's very attractive. You get out of the messiness of piecing together a process and get into just one process. It's cohesive and it is easy to troubleshoot, which in a lab operation, it is key in order to keep our operation flowing properly. QIAGEN really is a collaborator at this point. They can help us. They can help us do better. And that's what, ultimately we thrive for.
Thank you, Mr. Asakari, for your informative presentation. A quick reminder for our audience on how to submit questions. simply type them in the Q&A box, found by clicking on the green Q&A button at the lower left of the presentation window. We will get to as many questions as time permits. So without further ado, let's hop in and ask the first question. The first question asks, how do the multiple flow cell configurations work?
Yep. So, Yi, or do you want to take this, or I can briefly jump in?
So, yeah, the way the flow cells work, as Salim showed earlier, there are 3 flow cells in one GeneReader system that can each be run individually, simultaneously, or in a staggered fashion. And as Salim explained, this offers the greatest level of flexibility for laboratories, especially those with some fluctuation in their sample volume. So you can choose to run 1 or 2 or 3 today, and then tomorrow, if you only run 1 or 2, you could run the other 2 or 1 flow cell in a staggered fashion.
Excellent.
Okay, we can move on to the next question.
The next question asks, where can I find more information on the system? Can I see a demo?
The answer is absolutely. You can find more information on GeneReaderNGS.com, and you can absolutely see a demo as well. There is a contact us button on the website, and you could also feel free to reach out to any one of us here. And you can also, we will also be attending a number of upcoming conferences such as US CAP, ACMG, and Berlin, as I was referring to earlier. And you'll be able to see the GeneReader up and close and get a personal demo there.
Excellent. Thank you for your answer. Our next question asks, will there be GeneReader report template be provided to reflect the recent guideline standards and guidelines for interpretation and reporting of sequence variants in cancer?
If I understood correctly, I think the question is asking whether the guideline information will be updated and that will be reflected in the report. And the answer to that question is absolutely. So the database is actually continuously mined and added to and modified to, on an ongoing basis. At QIAGEN, we have a team of content curators whose sole job is to do just that. So any information, new information that comes in, whether that's a modification of a guideline information or it's a addition or deactivation of a clinical trial, that will be captured in just about real time and reflected in our knowledge database. So when a tumor sample, when a report is run through the database, the information you get in that report will reflect the most current information that's been updated in the database. And in your laboratory, because the interpretation piece will be web-based, you will have automatically real-time connection to the latest and greatest in our database.
Okay, thank you for that answer. Our next question asks, how do you determine the appropriate way to interpret, report cytogenic changes that are traditionally determined by FISH? Is this problematic for companion diagnostic?
Salim, you want to take that one?
Sure. So the FISH, is especially based on fusion detection. And fusion detection we enable through an RNA sequencing application, actually. And in a way actually that does not have to sequence the entire RNA, how would you say, the entire expression of the RNA molecule, but is especially tagged towards the beginning, which already shows whether there has been, for example, a skipping event or not. So and with respect to the actual what that means for CDX, well, I mean, the easiest way to answer that is probably to deep dive into this technology a little further and show you how this is actually being accomplished, and then check what the requirements would be, and then just take it from there.
Thank you so much for your answer. We have another question coming in today, and it asks, this looks like a very interesting platform. Does this enable harmonization of results from pharmacogenomic test assays?
Yi, do you want to, or I can give it a brief shot? So thanks for confirming the platform design, let's say, and harmonization of results is exactly what we want to achieve here. So I'm glad to hear that. With respect to pharmacogenomics, we have been looking into this. There is no standard. There's no assay actually that we have developed so far, but depending on, again, what the requirements here would be, it's always interesting to hear this back, from the expert labs, and we of course are always looking for fruitful companions here to develop something together, or at least gather the requirements from the expert labs. So again, here, if you would like to discuss this further, this would probably be the best way to find out if and how we could realize such an application. Yi, anything to add?
Yep. No, I think that's it. So, obviously at the moment we do not have an application that automatically combines, or enables the pharmacogenomic test. But that's not to say that, that's something that we should explore, and we definitely welcome reach out from people who might be interested in this.
Excellent. Thank you so much for your answers. Looks like we're running a little close on time, so we'll ask one more question today. How many samples can you process?
Yeah, so one can process, depending on the panel, for the pan-tumor panel, let's call it, the hotspot panel, one can process up to 16 samples per flow cell times 3, so up to 48 samples at the same time. But again, with the flexibility, you don't have to run all 48 at the same time, and you can choose to run just in one flow cell at a time. So up to 16. Minimum you can run one, of course, but a maximum 48 with a lot of flexibility in between.
Thank you for your answer. I would like to once again thank Dr. Kong and Mr. Asakali for your presentations. Dr. Kong, do you have any final comments you'd like to leave with us today?
I just want to say that, thank you again for all of your time and attention. We know GeneReader is a new system, and we definitely would welcome any further interest and for any further questions. And I know we didn't get time to go through all of your questions, so I welcome you all to reach out afterwards. Thank you.
Excellent. Thank you. And Mr. Asakali, do you have any final comments you'd like to leave us with?
Just also, we would like to extend the thanks, and I really like the type of questions that came in. So it really shows, it's a pity that we can't go through all of them. So if you'd like to contact us later on, we're very happy to continue the discussion.
Thank you so much. I'd like to thank the audience for joining us today, and thank Labroots for today's educational webcast, and QIAGEN for sponsoring the webcast. This webcast can be viewed on demand through May 2017. Labroots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. This is Christina with Labroots, and we look forward to seeing you here next time at labroots.com. Goodbye.